Today we will discuss about the microbiology of cheese. We know that cheese is a fermented milk product and it is now very popular in India also, because the young generation is very fond of pizza pie and different varieties of cheeses are used in making cheese pizza, and also cheese is also eaten as a along with salads. So, cheese can be defined as the curd of milk separated from the whey and pressed into a solid mass. Cheese is the curd or substances formed by the coagulation of milk, and milk may be from different species, that is cow milk is used in the world largely for cheese making, but specialty cheeses are made from sheep, goat or buffalo milk. Like cheddar is the most important variety if we look at the volumes of cheese produced world over. This is mainly prepared from the cow milk, whereas mozzarella is prepared from buffalo milk. Curdling is generally caused by rennet or similar enzymes in the presence of lactic acid produced by added or adventitious microorganisms. Rennet is an enzyme which comes from the stomach of the calf, but nowadays, as I said, similar enzymes, these enzymes are available from microbial production or from the genetically engineered variety of enzymes. Curd is processed to remove a part of the moisture by cutting, warming and pressing. Salting also plays a role in expulsion of curd and also in the inhibition of microorganisms. Curd is finally shaped in mold and then it is further ripened. So, coagulation of milk is important and it is caused by different mechanisms. As I mentioned, rennet coagulation is the most preferred, and the major varieties like the cheddar and Dutch variety Gouda or mozzarella and the Swiss varieties, maybe Emmental or other varieties, blue cheese, camembert, these are all produced by rennet coagulation. Whereas acid coagulation is used for the soft varieties of cheeses or fresh varieties of cheeses, mainly cottage cheese or cream cheese or quark, and heat and acid are also used in case of ricotta or channa and paneer. Channa and paneer really do not qualify in the typical classification of cheese. There are different classifications of cheese, and based on moisture they are categorized into low moisture, where the moisture is 13 to 34%, or we call them as very hard cheeses because the moisture is very less. Parmesan and Romano are two examples of this. Medium moisture, that is around 34 to 45%, and we call them as hard or semi hard varieties, and majority of the cheeses, that is cheddar cheese or Gouda cheese, that is the Dutch variety of cheese, and Swiss varieties of cheese, Edam or Blue, or these are all under the semi hard varieties of cheeses. And high moisture or soft varieties, fresh mozzarella or blue cheese or brie cheese, which have moisture around 45 to 50%. Then very soft varieties are like the fresh cheeses or unripened cheeses, in which we have the cottage cheese, cream cheese and ricotta cheese. Another classification of cheese is based on ripening of the cheese. Internal ripening is very typical and is used for the cheddar and also for the Dutch varieties of cheese, where there is the surface is covered by a paraffin wax or by special films, and the cheese is held at particular temperature of around 5 to 10 degree centigrade for a long period to allow internal changes, and whatever growth of microorganisms takes place, that takes place inside the mass of the curd. And mold ripened cheeses or blue cheeses, in which species of penicillium are used, the spores are prepared and as inoculum, and surface growth as well as internal spikes are given where some veins are formed inside. So, they are also known as blue veined cheese, and unripened cheeses are the fresh cheeses like the cottage cheese or cream cheese and fresh mozzarella or ricotta, which are consumed without ripening. They have smaller shelf life, whereas the ripened cheeses have longer shelf life and they can be stored for a longer period. So, for making cheese we use starter cultures, and acid production is the primary function of the starter cultures. Then flavor is very important, and eye formation, that is the gas pockets, is important in particular varieties of cheese. Inhibition of undesirable microorganisms is caused by the action of acid, because many of these spilage organisms and pathogenic bacteria are not able to grow at the lower pH. Creation of suitable environment inside the cheese during ripening or maturation process is also very important function of the starter cultures. There are other functions of the starter culture, that is, it promotes coagulation of the milk by rennet through production of acid, and also it stimulates curd shrinkage and expulsion of whey, so that proper level of the moisture can be attained in the cheese. And suppression of growth of undesirable organisms, controlling the elasticity of the finished curd, that the curd has to be rubbery and it has to be having a elastic texture, which is promoted by the right type of starter organisms. Promotion of fusion of curd into solid mass during the cooking, and controlling enzymatic changes during the ripening. Lactic cheese cultures are the most important cheese cultures. They are also known as primary starter cultures or primary starter organisms. In the mesophilic category, where we have this optimal temperature below 30 degree centigrade or around 30 degree centigrade, group N streptococci comprising of the species from the genus Lactococcus are very important. These are gram positive cocci which ferment lactose to lactic acid, and they are all homofermentative and produce the L type of lactic acid, whereas Streptococcus thermophilus is the thermophilic coccal organism which produces lactic acid, and homofermentative lactobacilli belonging to Lactobacillus delbrueckii. There are two subspecies of this, Lactobacillus delbrueckii subspecies bulgaricus and Lactobacillus delbrueckii subspecies lactis. Then non-lactic starters are also very important in particular varieties, as mentioned here. In Swiss cheeses, propionibacteria are used which produce propionic acid and large amounts of carbon dioxide by utilizing the lactic acid produced by the primary organisms, primary starters. Leuconostocs are also added to mesophilic cultures, especially for making the Dutch varieties of cheeses, where they utilize the citrate. Small amounts of citrate are present in milk, and citrate can be converted into carbon dioxide. So, small gas pockets are emerge in the Gouda or Dutch varieties of cheeses. Brevibacterium leanans is a pigment forming variety of bacteria which is used for surface ripening and smearing the surface. So, the typical red pigment is produced by these organisms. Species of fungi are also important, especially Penicillium roqueforti and Penicillium glaucum for the blue veined varieties of cheeses, and Geotrichum candidum is also used for the soft varieties of mold ripened cheeses. Optimal growth temperature, as mentioned by me, mesophilic and thermophilic. So, mesophilic are having between 25 to 30 degree centigrade, and thermophilic are used mainly 37 to 45 degree centigrade. So, Streptococcus lactis or Lactococcus lactis and Leuconostocs, they are the major mesophilic organisms, whereas Streptococcus thermophilus and Lactobacillus species, there may be Lactobacillus helveticus, Lactobacillus lactis or Lactobacillus bulgaricus, they are the major thermophilic organisms. So, flavor production by the action of lactic acid and salt is important, in this case. We can see that how this action takes place, that is the proteins are broken down, fat is broken down, lactose is broken down into different units, and this results in the production of esters, different fatty acids, amino acids, carbonyl compounds, which are important in flavor production. So, cheese ripening, as I told you, comprises of fermentation of lactose, which is converted first to pyruvic acid and then to lactic acid if we are using homofermentative bacteria, or if we are using heterofermentative bacteria, then lactic acid, acetic acid, ethanol and carbon dioxide are also produced simultaneously. Proteins are degraded to their building blocks, that is amino acids, and these amino acids are responsible for typical flavor. Fats are broken down to different fatty acids, and fatty acids also impart the typical flavor. Microbiological changes take place in different varieties of cheese. The ripening mode is very important, and hard and semi hard varieties are internally ripened. They discourage surface growth, and there is limited proliferation in the mass of the curd, that is inside the cheese mass. Soft and semi soft varieties are normally surface ripened, and smearing is used with typical bacteria, or typical strains of penicillium are used for making the surface ripened cheeses and mold ripened cheeses. There are different factors which help in the cheese ripening or which affect the cheese ripening, that is acidity and pH. Right type of acidity is very important for creating the environment. Similarly, moisture, if the moisture is high, then it encourages the growth of butyric acid bacteria, and salt concentration is important in inhibiting large variety of spillage organisms, and ripening temperature is an important factor in promoting the growth of the desirable organisms and bringing about the changes. Required storage of this curd at the suitable temperature, so that it gets transformed into the finished product by the ripening agents, that should be ensured. Temperature varies depending on the type of cheese, and period of ripening also varies. We can see that mozzarella and some of these cheeses are requiring very short maturation time, whereas extra mature cheddar may even take like more than 2 years. Parmesan also takes longer time. Higher ripening temperature is sometimes used for accelerated ripening. This brings down the total time taken for ripening, but sometimes the undesirable changes to body, texture and flavor will take place. Then the microbiological defects appear due to several reasons, and improper moisture content in the cheese is one of the important factors. It disturbs the relationship between fat and casein level in cheese, and the aging requirement or storage conditions. Usually related too much fat and too much moisture or too little casein, then it becomes pasty, or excessive proteolysis can be caused. Improper pH, or that is rate and extent of acid development, is a factor that affects the physical attributes of cheese. Poor quality of ingredients also affect the body and flavor and texture. Our microbial contamination is one of the most important factors which we can avoid by using good quality milk and proper sanitation in the cheese manufacturing facility. Simultaneously, the storage conditions are important, because poor storage conditions will compromise with the sanitary standards and contamination of the curd will take place. I have shown common defects in cheddar cheese here, and these are like development of openings. Cheddar is supposed to have a closed texture, but mechanical problems, that is disturbance of the curd or development of too little acid that allows the growth of undesirable bacteria, or pressing if it is not done properly. Too fast processing can be avoided, or lack of pressing can is also results in the open texture. And cold curd during pressing, that is the right temperature, or when it is hot during cooking, it should be immediately drained and pressed. High salt will also lead to the inhibition of microbial growth, and right quantity of salt should be ensured. The microbial spillage is caused by both bacteria, by yeast and molds. Many organisms, spillage organisms will grow, and they change the manifest itself in the form of visible growth causing discoloration, or bringing about changes in the degradation of polymers like fat and proteins by producing off flavoring compounds. Non-starter organisms play a very important role in cheese microbiology, and these are secondary organisms which can survive mainly pasteurization and are present in the raw milk itself. In this, the thermoduric streptococci are important, lactobacilli are important, lactobacillus casei, lactobacillus plantarum and lactobacillus brevis are well known to be very important in the overall ripening process in case of cheddar cheese. Similarly, micrococci are also heat stable, they can also survive pasteurization, and they contribute synergistic growth of lactic streptococci in cheddar cheese and non-lactic starters in surface ripened cheeses. Pediococci also sometimes become very important in flavor production, whereas gram negative rods like Pseudomonas and Achromobacter have also been found, though these are not largely considered as undesirable, but sometimes they are also helping in the enzyme production during ripening. Anaerobic spore formers or clostridial species, also known as butyric acid bacteria, produce large amount of butyric acid and gas, and these are major organisms of concern in the spillage of cheese. Any cheese milk having large numbers of these anaerobic spore formers is to be avoided for cheese making. Enterococci like Streptococcus faecalis or Enterococcus faecalis also produce lipases and proteases, which are helpful in the degradation of fat. Certain species of yeast, Scleromyces, Marxianus, Dibariomyces hansenii, Geotrichum, Candidum and Pichia species, and also different molds, Aspergillus, Cladosporium, Geotrichum, Mucor and Penicillium, they can cause surface growth during the ripening. That is why mold control in the ripening room is very important, and the ripening room should be maintained properly by fumigation and maintaining the relative humidity properly and ensuring good sanitary conditions in the ripening room. That otherwise the surface growth of these molds can cause different problems. Cheese also develops gassy defects, that is the cheese blocks will become bulged due to the presence of gas inside, and this is caused by different gas producing bacteria. Coliforms, if they come as contaminants, coliforms should be killed by pasteurization. So, they are very rare in the pasteurized milk. If the milk is under pasteurized, then these coliforms can survive and they will cause gas production during the initial stages itself, and this is known as early blowing. Similarly, if leuconostocs are in large numbers in the starter cultures, if there is a strain imbalance, or if the lactococci are inhibited, then leuconostocs may multiply more and they can produce large amounts of gas, causing early gas defects. Heterofermentative lactobacilli, unlike the homofermentative lactobacilli, also produce acetate, ethanol and carbon dioxide along with lactic acid, whereas the homofermentative on the other hand produce only lactic acid as the major end product of lactose fermentation. So, Lactobacillus fermentum, Lactobacillus buchneri and Lactobacillus brevis are known to be facultative heterofermenters. Lactobacillus curvatus is also important, and these can produce gas in the cheese which is undesirable. We also experience large amount of gas production at later stages. So, that is known as the typical blowing defect in cheese, and this can be both early or late blowing. Late blowing is of major concern, whereas early gas is normally due to coliforms or yeasts or due to leuconostocs. Late blowing defect is, as I told, it is the most dominant type of spillage of cheese contaminated with either anaerobic spore forming Clostridia, that is Clostridium tyrobutyricum or Clostridium butyricum, or propionic acid producing bacteria, which can utilize the end product of the starter organisms, that is lactic acid. These organisms convert the lactic acid into butyric acid and carbon dioxide, whereas the propionic acid bacteria convert the lactic acid into propionic acid and carbon dioxide. Heterofermentative lactobacilli or other heterofermentative lactic acid bacteria are also sometimes responsible for the late blowing. Clostridia, as I told you, they are heat stable because they produce spores and they are not killed by pasteurization. So, the cheese milk, which is raw milk, should be tested for the presence of these anaerobic spore formers, and if the count is high, then such milk should not be included, or the spores should be removed by physical process like bactofuse, that is centrifugal method, or filtration method. The gases produced by these butyric acid bacteria, they create typical blowing defect and large openness in the cheese interior in hard and semi hard varieties of cheeses. This may occur after a few months when pH starts increasing, because at low pH these anaerobic spore formers will not grow, and at higher pH they are activated and stimulated to grow. Propionic acid bacteria are used for the Swiss type of cheeses, but they may come as contaminants sometimes, and they may also utilize lactic acid, produce carbon dioxide and water. This carbon dioxide will produce the gas. In case of Swiss variety of cheese, the excess production of gas will result into defects where the eyes will not be in proper shape. Similarly, yeasts also cause, because they can utilize lactic acid and they can help in raising the pH and thus leading to the spillage of cheese. Many molds along with cheese are also able to grow at low pH and low temperature, which is used in the ripening process. So, these hygienic conditions, if they are maintained, then yeast and molds can be taken care of and good quality of cheese can be prepared. So, in cheese microbiology, mainly the starter cultures are important and the spillage organisms like anaerobic spore formers, propionic acid bacteria or enterococci, and also the yeast and molds should be checked properly. The ripening rooms should be maintained in good sanitary conditions, and raw milk should be selected for good microbial quality. Thank you.